Hello, welcome to some video notes on topic 3.1. And so for topic 3, we'll be getting into genetics, and we'll be talking about how gene expression is going to affect the phenotype or the physical characteristics of an individual. And we'll also be talking about chromosomes and meiosis and the effects of mutations. And we'll talk about certain specific genetic diseases that you have to be aware of, what type of mutation causes them, what type of problems does the disease cause in the individual that has it, and what is the inheritance pattern of those genetic diseases. So this first series of notes, we're going to be getting into some specifics about what a gene actually is and what are different types of mutations that can occur inside of a gene that can lead to a genetic disease.
So you get nothing. You lose. Good day, sir.
So starting off with some key vocabulary, first off, a gene is going to be a specific section of DNA that is going to code for a specific characteristic, and it will have a specific position on a specific chromosome. So for example, if you're thinking about hair color, there is a gene that determines if you have, lighter colored or darker colored hair. Whether it's going to be straight or curly would be another gene. So these different properties of your hair come from different genes. The location or the loci of these genes are all exactly the same for all human beings. So if it was chromosome number 5, about halfway down on the upper arm of the chromosome, if that was where the gene for hair color is, everyone's DNA would be the exact same locus. Now, there are different versions of genes, however, and we're going to be looking at, when we look at different types of versions of genes, which we call alleles, we get different types of inheritance patterns, and that's how we get some traits being more dominant over others, and while we have darker colors being more dominant than lighter colors. And so that's going to lead into some other topics that we're getting up in the overall category of topic 3. But for now, just talking about the idea of a gene. So it's this inheritable factor. Also, genes have to be inheritable, so it has to be that your genes came from your parents, right? And your genes will move on and go to your offspring, right? And so chromosome is basically, if you remember back when we talked about mitosis in topic 1, that's the condensed form of the DNA. Inside human beings, we have 23 chromosomes, and we have 2 copies of each chromosome because 23 chromosomes came from your father and 23 chromosomes came from your mother. So on a specific chromosome will be the DNA that's going to be used for making a protein, and so that one section on a section of chromosome, that one section of DNA, that is our gene. That's the one that controls that specific characteristic. And then the different versions of genes that exist within the human population, within the human species, those would be the alleles. So you could think of genes versus alleles. Genes give us a very specific type of trait, and alleles would be different versions of those traits. So I could say, you think of this in terms of like desserts, right? There are different categories of desserts. There are ice creams, there are cakes, there are pies, there are biscuits, right? And so within those different categories, you have variations in themselves. So within ice cream, you have different flavors. You have chocolate and vanilla and, rum raisin, stuff like that. So when we're talking about different genes, right, we're talking about different traits would be those different categories of desserts. And then when we talk about alleles, we mean within a specific category, all the different versions that exist inside of a population that are going to interbreed with each other. So for example, we could think of the hair color example I had earlier. That would be a gene for hair color, that's the trait. But there are different versions, different alleles for hair color. There's blonde hair, there's kind of auburn hair, there's very, very black hair, right? So there's all these different combinations that could occur to create different types of hair coloring. Okay, so again, regardless of the type of gene that we're focusing on, whatever gene we're referring to would always have a very, very specific location on a chromosome called its loci or its locus, right? Thankfully, because of the Human Genome Project, a 10-year kind of quest to sequence and identify all of the genes in human chromosomes and where they're all located, the ones at least that we have identified, we now know the loci of all genes that we know of and that we are currently studying. And so you can actually go on to internet databases and you can look up, a specific gene, and it will tell you exactly where that gene is located inside the human chromosomes, if you ever want to do research on that gene. Okay, so all individuals of a species are going to have the same genes, and they're all going to be in the same loci, right? But they're not all going to have the exact same alleles, right? If you have light-colored hair, your alleles would be slightly different than the person who has dark-colored hair, right? So there is always going to be a little bit of variation in our population because of this. And as we get into some other sections in topic 3, we're going to learn more about why variation is so important in a population and how we have evolved in order to make sure that we maximize the potential amount of variation every time we go through reproduction. Okay. So now we'll be, there's a little notice here talking about alleles of genes found in the same locus, but they could have different sequences, right? So the idea that blonde hair versus black hair, even though they're the same gene and the same loci, being different alleles, they of course would have different base sequences. They would have different orders of the A, T, C's, and G's, causing the proteins to be slightly different, causing the hair color, therefore, to be slightly different. Blonde hair versus black hair, there's going to be differentiations in coloration. Okay, now some interesting things about the genome, about genes, when we think about complexity inside of living things. So we said that human beings, for example, we had those 23 chromosomes in 2 pairs, right? So 46 in total. But if we were thinking about the number of actual genes that are present inside of a human being, we've identified a little over 22,000 genes that are used to actually make a human being, right? If we compared us to a virus, probably the smallest collection of genetic material that might actually do something, that would be a virus. So if we look at the influenza virus that can, cause you to get sick, very annoying virus, right? It only has about 11 genes in order, depending on the strain, but it's normally about 11 genes. Human beings have over 200 different types of cells, right? So over 200 types of cells and over 200 or 22,000 genes, where a virus is not even considered a living thing because it doesn't have all the characteristics of a living thing, only has 11. So there's a trend that we see, particularly with animal cells, right? This increasing in complexity correlates to an increase in the number of genes necessary to make that complexity. If we look at a bacterium, right? Bacteria cells, prokaryotes like E. coli, we see a jump up to 4,000 genes necessary in order to keep a bacterial cell functioning. But again, a very, very small number of genes compared to something more complex like a human. If we look at an insect, a fruit fly, 14,000 genes, right? If we look at a bird like a chicken, 16,000 genes. So as we're increasing in size, as we increase in complexity, there's more differentiation in the types of cells, the types of tissues that are necessary in order to keep that organism alive. And so we would expect there to be more and more genetic information required in order for that thing to be stable and to be a living thing. And so we would see an increase in complexity in the number of genes necessary as we go from something very simple to something very complex. Now, this is really, really great for animal cells. However, this rule does not apply when we think about plants. Here, grapes. Grapes, you would argue, are not really as complex as a human. They don't have nearly as many systems or organs, as a human being, a grapevine plant versus a human. Yet they have over 30,000 genes in their genome. So with plants, it's a little bit different because, as we're going to talk more as we get through topic 3, and we learn about the effects of mutations, plants don't really get affected by dramatically increasing the number of genetic information as they go from one generation to another, where animal cells actually do. So I'll give you an example. In human beings, there are 23 chromosomes, and there are 2 pairs of each. That means you should have 46. A very famous genetic disease that we're going to learn more about as we go through topic 3 is trisomy 21, or also known as Down syndrome. Down syndrome will lead to physical and mental limitations as a human being, and this is because you have one extra chromosome number 21. So instead of 46 chromosomes, you have 47 chromosomes. When it comes to animals, it seems to be the trend that having more genetic information is actually really bad for you, and more often than not, you don't even get to survive if you have extra copies of chromosomes or even whole extra sets of chromosomes, or what we call polyploidyism, having multiple sets of chromosomes, like having 3 sets versus 2 sets versus 4 sets versus 2 sets, right? With plants, however, that actually seems to be a benefit for them. Not only can they survive if they have extra amounts of chromosomes, but they actually tend to thrive and do better by having extra amounts of chromosomes. Give you another example, if you think about wheat that we use inside of our bread, right? The modern wheat that we use is hexploidy. That means that it has 6 times the amount of chromosomes than when it originally existed back when it was first being used for agriculture. Through controlled breeding, we've kind of crossbred and we've produced this new modern version of wheat that is 6 times the amount of chromosomes than the original ancient version of wheat. And by having more chromosomes, it's actually better for the wheat. It survives in drier areas, requiring less water, it grows faster, and it produces even more grain, which is what we would want because we're using it as a plant that's going to be used for agriculture to make food for us. So this rule that we will see with the number of chromosomes and also the number of genes doesn't really apply to anything that is based on plant cells, because plant cells have this adaptation that more genetic information is actually better for them. So something like grapes, or as you'll see later, ferns, can have tons of genetic information, but they really aren't as complex maybe as you would think compared to an animal cell. Okay. So to remind you again about our chromosomes, we did talk about chromosomes a little bit back in topic 1 and a little bit more in topic 2. And to remember, this idea of a chromosome is what we have when the DNA is going to be bound to proteins, which are called histone proteins. And it's going to wrap around those histone proteins, very similar to the way that thread wraps around a spool, right, in order to keep it nice and organized. And that creates what we call a nucleosome, which is a strain or a string of histone proteins wrapped around the DNA. And so then those nucleosomes will condense even more to create a fiber, and then those fibers can coil in on themselves to create a coiled fiber, and then those coiled fibers can coil again on themselves, creating what we call a supercoil, and then supercoils can coil again even further in order to create the physical version of the chromosome, the little X shape that a lot of people think of when they think of chromosomes. And so we're basically just taking DNA, wrapping around these proteins, and then wrapping it around itself in order really for it to take up less space. If you remember, the DNA is 2 meters in length, even though it's about 2 nanometers in width. It's very, very long, and so the diameter of a nucleus of a cell is actually very small, so in order to help it take up less space, DNA that isn't being used is going to be condensed into this kind of heterochromatin and euchromatin forms, where it's either tightly bound and it's not being used anymore, which would be our heterochromatin, or it's a little bit loosely wrapped around these nucleosomes and it can be used for protein synthesis, which would be the euchromatin. So remember, this is all stuff coming back from topic 1 and topic 2. And then if we want to go through the process of cellular division, like we saw with mitosis, we need to organize all the DNA into even more compact structures that we call chromosomes in order to easily move them around the cell. And so when we're talking about different genes and alleles, and we're talking about chromosomes, that's what we're referring to, is these tightly packed sections of DNA in order to keep them all nice and organized inside of the cell, and then later around the cell, or inside the nucleus, and then later around the cell when going through processes like mitosis and eventually meiosis, which we'll learn about in topic 3 as well. Okay, and so the number of chromosomes varies from species to species, and as we look at complexity, just similar to genes as well, there are more genes as you get more and more complex of an organism inside an animal cells. When we talk about chromosomes, it's the same thing. We see more and more chromosomes as we gain larger and more complex beings. And so the main reason why we're going to know everything that we know, and we're going to get into in topic 3, the main source of information is really coming from the Human Genome Project, which I said was 10 years, is actually 13-year project, where scientists all over the world were basically being tasked to help sequence the human genome, taking all the chromosomes one by one, different sections of the chromosomes one by one, and cross-referencing and putting all of their sequences into a database, and then using supercomputers to kind of process the areas where the information overlaps and kind of builds a solid puzzle of what is chromosome number 1 for a human being, what is chromosome number 5, for a human being, what genes do we find on these chromosomes, and what's the sequence, the normal sequence of these genes, right? There are different alleles like blonde hair versus black hair, stuff like that, but for example, the gene for ATP synthase. We need ATP synthase inside the mitochondria to make ATP. And the ATP synthase for human beings should be the same for everybody, right? So what is the sequence for the gene for ATP synthase? And where is that gene located inside of our chromosomes? So that was the goal of this Human Genome Project. And it took a good amount of time, but because of people in different labs working all over the world and sharing all this information with this kind of this open database, it was done fairly quickly. We were able to sequence all of the human genes. All right, and so now we know the full genome. And when we say genome, we mean all of the genetic material. So we know inside the DNA, we know every gene, but we also know the non-coding sequences as well, the sections of the DNA that aren't really used for anything, that are just there as extra filler, as buffer for, again, some of the mutations that we're going to be talking about, a way to kind of protect us against the effects of some of these mutations. But we really know of a lot of information about the DNA. And that really comes down from us using sequencing techniques, right? So things like PCR, which we've already talked about a little bit before, about making many millions and billions of copies of a section of DNA in order to get a very, very large pure sample, and then using it in order to go through various types of tests, using automated computers to go through the sequencing process, using basically fluorescently dyed nucleotides, ones that give off different colors, and using a computer and a really fancy laser to kind of read the DNA by looking at the colors that the DNA is going to produce. And so then the computer can interpret that and put that in the correct order through these phospholabeled nucleotides, the A, T, C's, and G's. All right. And then, of course, using digital cameras and all this advanced technology to identify all these dyes, and computers to assemble these base sequences together. And so for the HL students, all of this will sound a little bit familiar, and you'll have some more detail about how sequencing is done. But for the SL, this is just some general information to explain why we know what we know. And it's decades of research is backing up what we know about these sequences. So because of all this advanced research that we have, we can give you some really specific examples of where the genome can go wrong, or what we call a mutation. And so one of the mutations that we're going to be learning about in topic 3 that's really important and really well studied is sickle cell anemia. So looking at these two sections here, we have we're going to go through the process of what happens when a mutation occurs, and how a single mutation can cause a major change in the properties of a cell, and therefore the properties of an organism. So if you wanted to practice this idea of stuff we learned back in topic 2, if you think of the sense, the antisense, and the mRNA. Obviously, you don't have your amino acid chart in front of you, but if you did, you could go and find it if you wanted to practice that as well. But you can pause the video and you can write the antisense and mRNA sequence that would go with these two different chains, right? So if you remember your base pair sequencing and all this stuff, remember the sense and the antisense rule. So the sense is what actually gives us the protein, but because we're base pairing, when we use RNA polymerase to make mRNA, we can't use the sense chain. We have to use the antisense chain when we go through the process of transcription. So you'd have to be able to know the antisense chain, right? And then that would be used in order to make the mRNA sequence and then lead to the amino acid sequence, right? So going through the process of transcription, our antisense here, if we're looking at our normal hemoglobin, which we use HBB as the gene shorthand for hemoglobin, is the HBB gene, okay, on chromosome 11 here. So if we had the sense of CCT, AGA, GAG, we would have GGA, CTC, CTC as our antisense. And if we go through the process of transcription, of course, the sense And transcription, the mRNA would be exactly the same, except all of the T's would be replaced with U's because uracil is used in RNA instead of thymine. So instead of CCT, we'd have CCU, and of course GAG and GAG, right? Then we go through the process of translation, and if you had a translation table in front of you or on your test, you could easily look up the sequence, breaking it down into these little codons. You could translate each one of those codons into the amino acids, and you would find that you would see proline, glutamic acid, and glutamic acid. And that would give you, or part of this is necessary for making normal hemoglobin. That's not the only thing that makes normal hemoglobin. It's not 3 amino acids, but we're only going to focus on the one section where the mutation occurs. But there are other amino acids that are necessary to make hemoglobin. We're just focusing on one area. So that's normal hemoglobin, right? If we look at sickle cell anemia, we're looking at a mutated strain of, sorry, a mutation causes a mutation or a mutated strain of this allele, of this gene, sorry, creating an allele, a different version of the same gene. And so this would be the sickle cell gene. And so this is caused by what we call a single base substitution mutation. So a single base, one base in the sense chain, has been changed into a different base. So instead of a G, we have a C, or instead of an A, we have a T, right? Something like that. Or instead of a C, we have an A. It could be any base was substituted for another base. So when we look at the mutant strain, right? And we have CCT is the same, and the last GAG is the same, but the middle GAG is not what it's supposed to be. It is a GTG. Right? And so we go through the process of the antisense and the transcription and the translation, we don't end up with the same amino acid sequence in the end, so we don't end up with the same protein when this process is all done. So we go through the antisense. The antisense is going to be GGA, CAC, and CTC. And then we use that to go through transcription, which means our mRNA is going to be CCU, GUG, and GAG, right? And so we have a different codon when we're going in through translation. So if we look at our translation table, instead of a glutamic acid, we have proline, valine, and then glutamic acid. And now this is a big problem because proline, they're coloring it blue because it is a polar amino acid. Valine, they're coloring in red because it is a nonpolar amino acid. And so if you remember back when we talked in topic 2 about the forces that determine the tertiary structure of a protein, how a protein folds into its three-dimensional structure, right? You have positive and negative forces, you have disulfide bonds, and one of them is also the hydrophilic and hydrophobic properties of the amino acids. And so since valine is now is hydrophobic and it's now in there in our protein chain, the hemoglobin protein is not going to fold properly. And since it doesn't fold properly, it takes up kind of more space than it needs to. And as the hemoglobin gets stacked inside the red blood cell, not really taking the proper shape, the red blood cell ends up looking like this. It has a sickle or a half moon type of shape instead of being a nice circular shape like it's supposed to, right? So this sickle cell disease really means that the red blood cells are sickle in shape, and that causes a lot of health problems. Because of the sickle shape, they're more likely to get caught in the circulatory system, in the capillaries, which are very, very narrow. So it decreases the efficiency of blood flow, increases the pain in the joints. It causes the person needs a lot of rubbing their joints and massaging their joints in order to help circulation through the extremities like their fingers and their toes. There's less surface area, so that also decreases the efficiency of gas exchange, and it also means that the hemoglobin itself is also less efficient at holding onto oxygen. So somebody with sickle cell anemia is going to have problems with transmitting oxygen around their body. And if you have problems with transmitting oxygen around your body, that then leads to a whole other bunch of health problems because so much of your body is dependent on your oxygen levels. All these other things that are happening inside of your body depend on these oxygen levels. And so here, looking at an example, very well studied through this, the Human Genome Project, we now know exactly what causes sickle cell anemia. It's this single base mutation in a very specific section of chromosome 11, which leads the normal HBB gene to become the mutant allele version of it, which creates sickle HB, or sickle cell, leading to sickle cell anemia. So again, looking at the idea of glutamic acid versus valine, if we look at their physical structures here, we can see that the very end of glutamic acid has a negative charge right here, so it's going to be polar, versus valine, which has only CH3 groups, or methyl groups. Carbon and hydrogen together, that's what creates our fatty acid chains, right? Because hydrocarbons are nonpolar. So valine is going to be nonpolar, where glutamic acid is going to be polar. And so because of this small change, we have a huge ultimate effect on our hemoglobin structure, which leads to a whole genetic disease and a lot of health problems for the individual if they happen to inherit this gene. So what we're leading into is talking about the different types of gene mutations. So new alleles can come from mutations, as we see here. Hemoglobin is one version of the globin gene for blood, and sickle cell hemoglobin is an allele. It's a different version of the same gene. Now, unfortunately, most mutations, if we notice them at all, are probably going to be bad. But every once in a while, we do get a beneficial mutation, and if we have a beneficial mutation, normally that sticks around in the population thanks to natural selection, and so it will carry on and continue to appear in the population, obviously increasing in number because it gives the people that have that gene a benefit. If it is a negative gene that causes some type of disease or some type of problem, normally it will disappear from our population after a certain amount of time because it's having a negative effect, right? But most mutations actually have no effect because there are little buffers that exist inside of the way our DNA is dealt with that actually decrease the effects of mutations. Because even though mutations can cause good things, more often than not they end up causing a negative effect. And so we actually want to protect ourselves through these buffers protect ourselves from the effects of these mutations. But before we get into that, let's just learn about the different types of mutations that can occur. So a mutation, when we talk about our definitions, is a permanent change in your DNA base sequence, right? And once it's in there, and it can no longer be corrected, right? So if you have some type of mutation that occurs in your sex cells, like your gametes, your sperm or your egg cells, and then those cells are then used for reproduction, and so that mutation is then in the starting DNA sequence of that individual, that child that you produce. That mutation is permanent. We cannot go into every single one of their cells and change all the copies of their DNA. There's no way of getting away from that mutation, right? So this permanent change to the base sequence, all right? So this can happen through lots of different things. It could be caused by mutagens, right? Things like carcinogens. Cigarette smoke actually just doesn't have to be cigarette. Any form of smoke, of high energy, high temperature burning material is a carcinogen. It can lead to mutations, and some of those mutations, if they happen in certain types of cells in certain areas, can lead to cancer. So we've already kind of talked about some of the effects of mutations. There are other mutations, however, that don't necessarily lead to cancer, and they might just change the structure of a protein. It could be a good change. It could be a neutral change in which there really is no change at all. Or the mutation might happen in a section of the DNA that's no longer being used. So it doesn't really matter if the mutation happened there because the DNA isn't getting used for anything. Give you an example. Red blood cells, right? The bone marrow cells that are producing the red blood cells inside of your bone marrow, they're really focusing on using the DNA that makes a red blood cell. You have DNA inside of you that are used to make nerve cells and lung tissue cells. If you have a mutation in a gene that's necessary for a nerve cell, but it happens in a red blood cell, then that doesn't matter because the red blood cell wasn't using that DNA in the first place. So that's why most often, if we are getting a mutation, we probably don't even know that it's happened because there's a small probability that it actually happens in an area of our genome that we're actually using in those specific types of cells, and then it needs to cause some type of positive or negative effect after even getting through the buffers that we'll talk about a little bit later about how we can protect ourselves from mutations. Okay. So the accumulation of mutations, though, in a population, in a species over millions of years, can be a really strong force and actually can have a huge effect on a species. And so when we think about the development of species and the evolution of species, that's almost entirely driven by mutations. And so mutations are not really going to have such a really strong effect on you in your lifetime unless you were born with a genetic disease, or you have a chance that you might give a genetic disease to your children. But mutations collectively that appear inside the human genome, right, the human race's collective DNA, that is going to accumulate over thousands of years and will have a substantial effect on where we go, where we will evolve into. Okay, so not all mutations end up causing disease, but some do, and we'll get into some of those mutations and those different genetic diseases as we go through more in topic 3. So even though DNA replication is really, really good at following base pairing, DNA polymerase is really good at its job, right? If you're an HL student, remember we talked about different types of DNA polymerase. There's DNA polymerase 2, which is actually a proofreader that scans the DNA after DNA replication has occurred to make sure that it did it correctly, and if it finds a mistake, it will fix the mistake. So even though there's all these little things done through DNA replication in order to reduce the chances of mutations, mutations always occur. There's too many base pairs, and there's too much of a probability that there will be a mutation at some point while going through the process of DNA replication. So we can get, even though there are environmental things like radiation and carcinogens that could cause mutations to occur, and do cause mutations to occur at a relatively consistent rate, we will always get mutations on some level because of DNA replication. The whole process is not perfect. So because there will be a mistake at some point, mutations are bound to occur. And at some point, if a mutation happens in a gene that really does get used in a very specific part of the body and makes that part of the body not function very well anymore, that's when it leads to what we call a genetic disease. So let's look at an example of like a base substitution mutation here. So a base substitution is one example of different types of mutations that can occur. We could have deletions where a whole, nucleotide or a section of a nucleotides disappear. There could be additions where extra nucleotides get added and so the DNA gets a little bit longer than it should be. There could be inversions where whole genes or sections of chromosomes get swapped around when they're not supposed to. Right, so there's lots of different types of mutations that can occur, but we really want to focus just on the base substitution mutation and what could happen if the base substitution occurs in a section of the DNA. Right, so we substitute one base for another. So here we've got a section of DNA, we've got an area for you to do the mRNA, and we have an amino acid chart here. And so we'd want you to practice using the amino acid chart. So I want you to pause the video and actually give this a chance, right? See if you can figure out what are the amino acid sequence, the mRNA sequences and amino acid sequences based on these two different DNA sequences because of this mutation at the end. All right, you give it a shot. Okay, so we look at our mRNA. Again, we would replace the thymine would not be used. We'd be using uracil. So we're base sequencing against that DNA. So we'd have UCG, GGC, and AAU. The mutation, because of the G at the end, has been changed. So instead of AAU at the end in blue, we have AAC. If we look at our amino acid chart, if you want to, there are lots of different amino acid charts, and so it's good to practice using the different types. So our first letter is here in the inner circle, the second letter is in this later circle, and then the third letter is in the outer circle. And so you basically just have to start with the first letter and then just move through them. So for example, we have UCG, so we start with U, then we go to C, then we go to G, and then we see that it's serine. And actually, because of the degenerate nature of the codon system, UCU, UCC, UCA, and UCG all give you serine, right? And so lots of codons can actually give you the same amino acid. So if we look and we go through this process, all right, we would see that this is a silent mutation. All right, we have serine, glycine, and asparic acid, right? Asparagine, yeah, asparagine for ASP. But because of the mutation, AAC still gives you asparagine, right? If you look at the chart here. So since you end up with the same amino acid sequence, the protein that gets produced is still the same, so it still functions the same, so there really isn't any effect to this mutation. So silent mutations are when we have, for example, a base substitution mutation or other types of mutation, but we don't have any actual effect. It doesn't do anything, either positive or negative. It's a neutral effect, right? And the whole reason why we're really good at protecting ourselves from mutations like this is because the genetic codes, the codon system here, is degenerate. So having so many different codons give us the same amino acid, or multiple codons producing the same amino acid, leads to the effects of mutations to be reduced. Because if we get the same amino acid sequence, we get the same protein, so we never really had a negative effect as a result of this mutation, so it doesn't really matter that the mutation exists. All right, if we look at another example here, we've got our DNA sequence, mRNA sequence, amino acid sequence for both of these examples. We've changed the location of the base substitution. Here, the CTC has been changed to CAC, and here at the bottom, the ACC has been changed to ACT. All right, so go again, pause this, go through this, and see what you get. All right. So mRNA sequence, right? Probably pretty good at that. And you probably recognize that this is the example that we talked about earlier. So this would be an example of a missense mutation. And so a missense mutation is when we've changed the protein to some new version of the protein. Normally it's a negative effect. It doesn't necessarily have to be a negative effect. It could be a positive effect. All it is that the mutation has caused an actual change in the shape of the protein, right? More often than not, a missense mutation is negative, but there also can be positive effects. It all depends on what happens when we actually produce that protein. An example that we're looking at here would be a negative missense mutation where we have sickle cell anemia, right? The glutamic acid has been replaced with valine. We look at the lower example, here's the mRNA sequence, right? And then going through the amino acid sequence, we have a very interesting situation that has occurred. We have a premature stop codon getting inserted into our protein. And so we would call this a nonsense mutation. And so nonsense mutation, basically, it doesn't necessarily have to be the addition of a stop codon, but most of the time it is the addition of a stop codon. Because the amino acid translation, or the mRNA being translated into amino acid sequence, stops much earlier than it's supposed to, it produces a shortened, useless form of the protein. And so in a missense mutation, the protein might not do its job very well, but it could still be functional as a protein. In a nonsense mutation, the thing that it's producing does not function at all. And so one example of this would be like citric fibrosis. And we'll talk some more examples in later topics about what citric fibrosis actually is. But because of a nonsense mutation, the effects, the physical effects of citric fibrosis are produced. Okay, so there's just different examples of how these mutations can affect us, depending on if they change the sequence, they don't change the sequence, or if they change the sequence enough that the protein doesn't actually function at all. So, again, looking at this example of sickle cell anemia, just kind of summarizing everything that we've talked about. Because of a single base substitution of a glutamic acid or glutamine being changed into a valine, it produces abnormal hemoglobin. This hemoglobin, we call it HbS, by the way. Adult hemoglobin, sometimes they call it HbA, sometimes they call it HbB, sometimes they call it HbN, right? There's different terminology that goes for it. I think HbN is probably the more common one, so that's N for normal, right? Normal hemoglobin, where HbS stands for sickle cell anemia, or sickle hemoglobin, and we always use the same thing, HbS, when we're talking about sickle cell. So because of that, it creates a lot of health problems. The number one concern is that it decreases the efficiency, or it decreases the amount of oxygen that the red blood cells can transmit, and then it also decreases the efficiency of the movement of blood through the body, because it's more likely for these sickle cell shaped red blood cells that get caught in our capillaries, and it causes a lot of pain and reduces the efficiency, the movement of oxygen around the body, which causes a lot of other health problems that the person will have to deal with for their whole life, because again, all of their red blood cells are going to be expressing the same gene, and so they will all have the same physical trait, and this will be a lifelong problem, which is what we think of when we have a genetic disease. We're talking about something that's in the DNA, so there really isn't a cure for it. There's only ways to help people deal with the symptoms, but we can't get rid of the disease. So you're probably wondering at this point, wait a minute, didn't he say earlier that if it's a negative mutation and has a negative effect, causing disease, causing some type of limitations in health, isn't it more likely that gene will eventually disappear, or the disease will eventually disappear, because negative things in our genomes, typically with natural selection, tend to fade away, right? Even though human beings are doing a really good job not letting ourselves be affected by natural selection right now. And you are right to think about that. When we think about sickle cell anemia, first off, sickle cell anemia isn't a new thing. It's been around for hundreds of thousands of years. It's a disease that has existed well into the past. It's just recently, in the last 60 years or so, we've really identified and know exactly what it is. But it's been around for a very long time. But why does it exist? Why didn't it just get bred out of the human population long ago? If you had sickle cell anemia, you probably had a really hard time running away from a predator or catching your dinner because you weren't able to transmit oxygen very well around your body. You probably had a hard time finding somebody to reproduce with if you did survive. So it probably would just disappear from the environment after enough time. Well, there are two things that are contributing to this, and we're going to learn, the first one I'll say is that it is a recessive trait. And we'll get more into why that is important a little bit later, but in another topic. But to kind of summarize it, recessive traits, because they are recessive, they can actually hide in a population pretty well, and people not aware that they have them. And so they could give it to their offspring, not realizing that they're giving them a faulty gene, like sickle cell anemia, because it is a recessive trait. Or not recessive, but it's codominant. So we'll get into that in genetics a little bit later. The other thing is that there actually is an advantage to having sickle cell blood depending on where you are in the world. And so if you look at the chart here, the highest areas of sickle cell anemia occurrence is in this region of the world, right? It does happen in some other parts of the world. It's not completely limited to just parts of Africa and the Middle East, but the highest occurrence is in Africa and the Middle East. And there's also a now growing number over here in Southeast Asia. And you'll probably notice that based on this little map here, that the red areas are also highlighting sections of the world where there is a lot of malaria. Now, malaria is a disease, a different type of disease. This is a contagious disease. This is one anybody can get, technically, caused by a prokaryote, a eukaryote, a paramecium, a eukaryotic cell called plasmodium. And so what plasmodium does is it will get into your body, and it will actually use your red blood cells to help complete its life cycle. Your red blood cells, and actually sometimes your liver cells, depending on how bad the case will get. Now, famously, malaria is the reason why mosquitoes are considered to be the deadliest organisms on Earth, because mosquitoes are what transmit malaria through a really complex life cycle. Mosquitoes will bite someone that might have a later form of the malaria life cycle, and it gets into their body. It doesn't really have a negative effect on the mosquito, because the mosquito does not have the same type of system that we do with the transmission of oxygen. And so it will grow inside of the mosquito, and then later when the mosquito ends up biting another individual, through the process of it exchanging some bodily fluid, the mosquito then gives that person, if they don't already have malaria, will give them the plasmodium cells, will infect them with the cells that can cause the disease. So these eukaryotic protists these parasites will increase in number by using your red blood cells to help them with their reproduction. And so your red blood cells start to die as their numbers start to increase. And eventually you get anemia. And if you get really, really sick and you don't get medication to help remove these parasites from your body, you will die because they will kill so many of your red blood cells that you won't be able to transmit enough oxygen to survive, right? So that's what malaria is, basically an invasion of these plasmodium cells that kill your red blood cells, leading to fever, shivering, vomiting, anemia, and then eventually you could have kidney failure, fall into a coma, and then you could die, right? So it is a disease that could be lethal. So that's why we say the mosquitoes are the most dangerous organisms on Earth. They kill more humans than anything else because they're transmitting this parasite to us. Okay, so the thing is that the reason why sickle cell anemia is an advantage is because malaria is really, really bad at infecting, or the plasmodium cells are really bad at infecting sickle cells. So if you have the sickle cell trait, if you have sickle cell anemia, you're actually going to be very, very resistant and practically immune to the malaria disease. So if you have if you have sickle cell disease here in the world, that's not good. That's not going to be good for you, and you're going to have a really difficult life. However, if you have sickle cell in these regions of the world, all right, over here as well, that's actually pretty good for you because you are going to be immune from one of the most dangerous diseases to face mankind by just happening to have these cells. You will have other health problems, but you will be protected from this very dangerous disease called malaria caused by these plasmodium cells, right? So that's where we see this trait staying around inside human beings, because in certain parts of the world, it's an advantage. And so it's actually better for you to have this disease, or have sickle cell anemia, right, this genetic disease. You're more likely to survive, and if you are surviving and you are fairly healthy, maybe you're more likely to reproduce, and so you pass this gene on to your offspring, and maybe they have sickle cell anemia, maybe they have a codominance, they have a mixture of normal blood and sickle cell blood, which is actually the best combination possible, which we'll talk about in some other topics, right? So some traits, even though they might be negative, they might cause diseases, they could be positive under the right circumstances. So when we think about natural selection, we think about this accumulation of traits over lots of time, we could think about sickle cell disease, right? Even though sickle cell anemia, sickle cell disease isn't great for other things, obviously it must have become an inheritable adaptation that occurred as a response to dealing with malaria. And as malaria increased in a region, someone with sickle cell anemia happened to be immune to disease, they're less likely to die from it, the more likely to survive and get to reproduce. And so gradually over many, many generations, sickle cell anemia becomes a very, very common trait in certain parts of the world because of this environmental factor selecting for it. And so this is really how natural selection occurs. And we're going to talk in a lot of great detail about natural selection when we get to topic 5. So there's a little bit of overlap between topic 3 and topic 5 because of the mixing of genetic information with survival of species and adaptations of species. Okay, so that's it for this first section of topic 3. If you have any questions, let me know.